Hello, everybody. First, I would like to thank the organizers for giving me the opportunity to tell you about one of the projects that I've been working on during my postdoctoral training in Jonas Theis Lab at Yale University, which is about 3' end maturation of a cancer-associated long non-coding RNA called MALAT1 that is dependent on formation of a tRNA-like structure that I will tell you more about in subsequent slides. You know that RNA triple helices have been found in different important biological processes, including catalysis, ligand binding, and RNA stability. The particular class of RNA triple helices that I'm interested in are found in RNA stability element, and one of them is found in human MALAT1 long non-coding RNA. MALAT1 and MEN-beta, both of them are nuclearly, but highly abundant nuclear localized long non-coding RNAs, which have RNA triple helices at their 3' end of their mature form. Both of these long non-coding RNAs at their 3' region, they have an E element, or which is a U-rich internal loop, followed by a genomically encoded A-rich tract, and then a tRNA-like structure. And as you know, both of these long non-coding RNAs are associated with cancer, and specifically for MALAT1, it's been associated with tumor progression and metastasis, and that's why it's been considered as a potential anti-cancer drug target. MALAT1 3' end maturation has been shown by the work of Jeremy Willits and David Spector's lab that is dependent on RNase-P action. The tRNA-like structure is recognized by RNase-P, which is cleaving upstream of the tRNA-like structure, resulting in formation of the mature 3' end of MALAT1. And if for some reason this maturation is skipped, the polyadenylated transcript will be formed, which is actually not stable, and it has low expression, which is not detectable with the NORM-BOT analysis. So the question is that whether the triple helical structure and tRNA-like structure can form at the same time before RNase-P cleaves, or the ENE has to wait until RNase-P cleaves upstream of the tRNA-like structure, and then the mature 3' end will form and eventually form a triple helical structure. When you look into the primary sequence of MALAT1 orthologs coming from different species, as expected, in those particular regions that are associated with structural elements like the A-rich tract, part of the triple helix, or in masked RNA, which is forming the tRNA-like structure, they are highly conserved. But surprisingly, when the tRNA-like structure is ending, there are about 7 or 8 nucleotides that are again highly conserved, and then you see significant drop in the conservation at the primary sequence level. When you look more carefully into this particular conserved 3' trailer sequence of the masked RNA, you see that this boxed nucleotide in black is fully complementary with this boxed region of the A-rich tract. So it's predicted that the tRNA-like structure, upon the hybridization interaction of these two regions, can form an extended stem with a single adenosine-involved nucleotide. What I did next was to find out what is the functional significance of this particular secondary structure, predicted secondary structure. And I designed some variants like mutant 1, deletion 1, and insertion, which in all 3 cases, it is predicted that the extended stem of the tRNA-like structure is disrupted. However, in deletion 2, the non-complementary region of the masked RNA trailer is removed, which should not affect formation of the extended stem with the bulge nucleotide. To study the maturation of MALAT1, I used intronless beta globin assay, which is based on the fact that when you remove the 2 introns of beta globin, the transcript stuck inside the nucleus and will get degraded. And by normal blood, you can see that the level of intronless beta globin in the absence of any stability element is basically undetectable. However, after introducing a triple helical structure like MALAT1 triple helical structure, the expression of RNA is detectable. So in those 3 variants, which the formation of this extended stem is disrupted, you can see that in all 3 cases, there is a significant reduction in the accumulation of the transcript. However, in deletion 2, which the predicted secondary structure is not disrupted, there is no significant change in the accumulation of the beta globin transcript. Next, what I wanted to study was like, since upon maturation of the transcript, the tRNA-like structure should be produced as well, I wanted to look into both maturation of the transcript and looking into formation of the tRNA-like structure at the same time. And I also introduced like two other variants, one called delta A, which is removing this single bulge nucleotide, and then with a compensatory mutation of delta U, basically recovering the formation of the bulge nucleotide. And I used the same internalist beta globin assay and northern blot analysis. And as you can see here, in all cases where the predicted secondary structure, meaning extension of the stem and a bulge nucleotide, is disrupted, both level of beta globin transcript and masked RNA drops simultaneously. But like, for example, here, when with the delta A mutation, the formation of the bulge nucleotide is recovered, you can see both production of masked RNA and beta globin accumulation is recovered. So this suggested before the action of RNase P, this particular conformational state has to form, and suggesting that the triple helical structure cannot form before the action of RNase P. So to provide structural evidence, I started to do structure probing using inline probing, which is developed by the Breaker Lab at Yale University. Here you can see like two inline probing gels for MALAT1 ENE only, which is not forming triple helix, and also MALAT1 triple helix. In each band in inline probing lanes represents a nucleotide in this particular structure, and the density of each band represent whether that particular region is flexible if the density is high and the band is bright, or if it's structure and rigid when the density is like reduced compared to flanking regions. So inline probing helps us to distinguish formation of the triple helix versus the ENE only structure, and there is a particular signature, and if I zoom in, for those two guanosines that are located in the urich internal loop, when you have ENE only, these two guanosines apparently forming non-canonical base pairing interaction with those two U's on the other side of the ENE, and that's why you see reduction in the intensity of those bands compared to the flanking nucleotides. However, when the triple helix is formed, these two guanosines are flipped out of the structure, and you can see the signature, which is significant increase in the intensity of those two bands compared to the surrounding nucleotides. Next, I designed several MALAT1 ENE plus A-rich tract plus masked RNA construct to study the conformation and folding of the 3' end region of MALAT1. Three of these constructs, I've already talked about them: wild type, delta A, and mutant 1. Two of them predicted to form no triple helix, and mutant 1 should form triple helix because the A-rich tract is not basically able to form this extension interaction with the trailer sequence. In mutant 2, the whole tRNA-like structure is disrupted again. It is predicted that the A-rich tract will be available to form triple helix. And finally, in true negative control, or TNC, the A-rich tract is actually swapped by the U-rich tract in the trailer sequence of tRNA-like structure. This should not affect formation of the tRNA-like structure with an extended stem, but would basically prevent formation of the triple helix. And when you look into the inline probing pattern, and specifically the diguanosine signature, you see that those three variants, like wild type, delta A, and true negative control, which are predicted to form no triple helix because of the formation of a tRNA-like structure with an extended stem, the diguanosine nucleotide show no significant intensity. However, in M1 and M2 variants, which the tRNA-like structure and formation of its extended stem is disrupted, both of them show significant increase in the intensity of diguanosine bands, which is the signature of formation of the triple helix. So our data, cellular and structure probing data, suggests that none of the previous models are correct, and indeed MALAT1 3' end should form a tRNA-like structure with an extended stem before the action of RNase-P, and after processing by RNase-P, the triple helical structure can form. And the reason we think this is very important is that the triple helical structure of MALAT1 is considered an anti-cancer drug target, and a lot of efforts has been going to identify small molecules targeting these triple helical structure in order to destabilize it. But this model shows that there is a conformational state before the triple helix is even forming that has perhaps been a better target. So with that, I would like to thank John and the whole STICE lab members for their help and support and funding sources. And thank you so much for your attention. I would be happy to take any questions if you have any.